Hello everybody, my name is Wang from Sherbrooke University, and today I will present my work concerning the IDH mutation that induces a neurotransmission shift in glioma. We know that glioma are the most aggressive adult human brain cancers, that they can be classified into three subgroups based on the IDH gene alteration and the 1p92 chromosomic aberrations. The IDH white-type glioma bears the worst survival compared to the IDH mutated glioma. In the other part, neurotransmission is the main brain process involving in many neuropathologies such as Parkinson's disease. Its association with glioma development via the GABA and glutamate neurotransmitters is known but not very well understood. The existing drugs targeting the neurotransmission system could be an interesting way to complement current glioma therapies. Our hypothesis is that GABA and glutamate-related neurotransmission could impact on diverse biological regulation in gliomagenesis. In our study, we try to discover this mechanism and to decipher its role in glioma development. We started our investigation extracted 661 glioma samples from the TCGA. We also extracted the GABA and glutamate related gene from the cake metabolic pathway database. Then, we performed supervised clustering that allows us to generate a heat map, and from this heat map, we will be able to generate to identify 4 specific distinct clusters. We then performed comparative bioinformatic analysis to characterize the nature of each clusters. Unsupervised clustering allows us to identify 4 clusters with different GABA and glutamate expression profiles. We named each cluster by NT1, NT2, NT3, and NT4 clusters for neurotransmission cluster 1, 2, 3, and 4. The NT2 clusters, in blue, was associated with the worst prognosis compared to the other clusters. This was explained by the fact that the NT2 clusters were mainly composed of IDH white-type glioma, whereas the NT3, and NT4 were mainly enriched in IDH mutated glioma. The NT1 clusters, for its part, was composed of a mixture of glioma types. When looking to the GABA and glutamate expression levels, we noticed that NT2 in blue was associated with the lowest expression of these neurotransmitters genes, whereas the NT1 in red was associated with an overexpression of these genes. NT3 and NT4 clusters show intermediate levels of expressions. Correlation analysis of these neurotransmission genes demonstrated a negative correlations with between the neurotransmissions and the immune process. We saw that there is a negative correlations between the cell activations and the cell populations proliferations related to immune cells. We also have a correlations between immune effector process. These correlations were also validated using CyberSort. We found that each cluster was associated with the specific human compositions, where the macrophage M2 populations were proeminent in the NT2 clusters, where the glioma bear the worst prognosis, and monocyte population were enriched in clusters such as NT1 clusters. In conclusions, in our study, we identified different clusters with specific and original attributes. These clusters were mainly based on the current classifications using the IDH and 1p92 codelations, but we observed that one of these clusters, the NT1 clusters, does not seem to follow this classification and was associated with the higher expression levels of these neurotransmissions. These expressions were also observed to be correlated with the presence of a certain immune cells, indicating a crucial role of this neurotransmission regulation on the immune cell recruitment in gliomagenesis. Our perspective will be to look at the factors explaining the relationship between the neurotransmission and the immune microenvironment. To do so, we will try to explain this relationship by doing interaction network analysis. So thank you for listening me, and if you have any question, don't hesitate to ask me or to send me an email. Thank you and have a good day.